Okay, I'm Ken Mitton, and this is basically a screenshot video here that will help you use the University of California, Santa Cruz Genome Browser and how to find genes of interest. So there's the address, and this is kind of the page that you land on when you first get to the Genome Browser. There's various tools. There's a lot of things you can explore more if you wish. But for today, we are going to just say that we want to find a gene of interest. And you can just cut right to the Genome Browser. And there's a couple places on the page to click and do that. And you'll get some kind of default view. Generally, it's going to load for you the latest build of the human genome sequence. So for those of you that don't realize this, there are different versions that are updated from time to time. The latest one is 38, also called HG38. There was a Human Genome Society, and there was also another consortium doing sequencing that were keeping track of genome sequences, and they had a different naming system. One's GRCH, the other one's HG. At this point, they kind of went right and started to use the same number, 38. And so they used to have different numbering. GRCH 37 was a different HG number that was much lower, but they kind of agreed to simplify things going forward. Basically, when you're in the browser, you're going to have a view that shows you an ideogram of a chromosome location. It looks like we're happen to be dropped on chromosome 11 here at the moment in my browser. And you see the P and the Q arm of the chromosome. If you generally aren't sure of what the P and the Q arm are on chromosomes, you can read a little bit more about that in any biology text. But basically, there are sections banding from traditional staining of metaphase chromosomes. And these locations have these little map numbers on the P arm. They start with P. The numbers are smaller as you go to the centromere. They get larger as you go to the telomere end. Same on the Q side of the chromosome. These locations were used for mapping genes in the past a little bit more than now, but they are handy landmarks for sort of knowing where you are in the chromosome. The view we have right now, it looks like I'm looking actually at the frizzled 4 gene, which is one that we work on, and it happens to be on chromosome 11, and it's positioned, and it shows you here Q14.2 is where we are sort of in the region of this chromosome. One thing we can do is find yourselves when you first get here the configuration tab, and some handy changes are you can make the picture image width a little bit bigger, and you can also increase the text size of labels that it's using. Go for something bigger, maybe even like 24, and you just submit that. Boom, and we go back to the view again, and things are pretty good. I might even want to make it bigger, and so if I want to do that, I will just continue to do that. But get things where it's comfortable for you to look at. So I got a little bit wider window now. All the labels are a little bit bigger. There's a lot of tracks that you may or may not have on, depending on if someone's used your browser to be here before, or if you have. There's all kinds of tracks that can be overlaid in the genome, and most of them we can turn off. And by hiding them and going to these controls, I'm going to do that. I'm going to leave the NCBI reference sequence on. I'm going to get rid of this gen code 44. I'm going to get rid of OMM alleles and disease genes. And I'm just looking through all of the different information bioinformatics tracks that I really just don't need to have on. Full conservation, these dark areas are showing you conservation of sequence between the species. Sometimes that's handy. For instance, we see that these thick exon regions of this gene are very well conserved among vertebrates. But let's get rid of some of these extra views that I just don't really need at the moment. Repeat masker I can get rid of. I think I turned off conservation. I'm going to get rid of that too, because I just want to really see the gene. I can hit anywhere. There's numerous refresh buttons. Anytime you adjust something, you can hit any one of them. And now this is basically a very simple view of a gene and its chromosome. So I can look for genes of interest here. And so if you want to look for a different gene, you can type them in. If you start to type, you will get pull downs of genes that are in there. I'm going for rhodopsin. The official gene acronym is RHO for human rhodopsin. Here's a reference based on a reference mRNA sequence. I can zap right there, and I go right to chromosome 3, and here's rhodopsin. I can zoom out a little bit if I want to see what's around it. And looks like I got gen code flipped on again. Let me flip this off. Refresh there. Ref sequence genes from NCBI are just for most genes there's a reference sequence that's been curated with some human knowledge overseeing it to sort of give a basic reference sequence for every gene. But there's where we are. We can zoom out quite a bit with genes and see the other genes that might be hanging around. And you can see there's plenty. Let's zoom back to rhodopsin again, a gene I've worked on for many years myself. There we go. Sometimes you can click on these labels, and as you click, it'll cycle through different views of the gene for you. You can always reset it just by going to look for rhodopsin again. Boom. There's rhodopsin. So it looks like it's going to want to pull up gen code 44 to show me the rhodopsin naming label, which is fine. We can work with that. Oops. Come on. Load for me, load for me. There we go. So when you get here, your gene may or may not be facing in a specific direction. So generally, if this chromosome's DNA is laid out, one strand we'll call the top strand, one is the bottom. Top strand, the sequence of the DNA is going from left to right. The opposite strand, it would be going from right to left. These little chevrons in this structure show us the direction of the gene. These thick regions are exon sequence, and the thin lines are basically intronic regions. Once this is all transcribed as a initial messenger RNA, it will be spliced, as you know, and all the sequence that originated from the exons will be spliced together, removing the intronic sequence. Do note that exons do not correspond one-to-one to coding sequence of an amino acid. That is a common misconception that I find even many new graduate students have sometimes. While it is true coding regions are within exons, not all of the exon sequence is coding region. And so this is the first exon of the rhodopsin gene. It's transcribed. The thickest part of the bar here is to show you that's coding region. So there's a start methionine at the beginning of this, start that progranine. This other medium-sized thick region is 5' non-coding sequence that will be in the RNA. And many genes have a rather long section of 3' non-coding sequence. And there might be control and trafficking signals in there that are used by the cell. There's a sequence in there that tells an enzyme system to put a poly-A tail on the end of this. And so these things exist. But here's the structure of a gene. I got 1, 2, 3, 4, 5 exons. And I know it's on the top strand oriented in this direction. You can flip this at any time by hitting this reverse button. And now it shows me it's reoriented the chromosome, and it's also reoriented the sequence. So you can see the sequence is numbered lower here, higher over on this side. And it's showing me the direction of the gene. So now I would know that in this orientation, the bottom strand is where my gene is oriented. So you might go to a gene and find it this way at first. If so, you can just flip it back around by hitting reverse, and you can go back to the way you want to see it at any time. Back and forth. There we go. So that's basically how you can zoom in a little bit, in and out on these genes, but that's how you get a picture of the gene. So if you want to actually have this structure and you want to get it as an image, you can go to, I think I can go to viewing here, view PDF. I think I want to do that. Let me get this right menu. Yeah, and in here you can download the current browser graphic in a PDF. And that would be the gene diagram that you've built and customized to look at. This other link will download a little picture of that chromosome ideogram. So if I go over the first one, it will give me this view, and I can download it. This will download, and it's a PDF. It's very handy because you can shove this into a poster, a slideshow, or your essay on Island of the Colorblind, for example. Although it won't be rhodopsin, it'll be a different gene, but you can then just download that file and you'll get the file. So save her in there. And mine automatically opened up in my preview app on the Mac, and there's the file. I have it on my desktop now that you can put in your essay, for example. The other link for the chromosome ideogram is here, and there's how you can get this picture again, and you can download it just the same. So that's how you can get to the structure of any gene in the Genome Browser. You can't really hurt anything playing with the browser. You can play around with the different controls and see what you get. It's really quite a nice tool to really get familiar with where the heck you are in the genome. Sometimes you might like another good view is this main, M-A-N-E. This view is another good mapping reference sequence for the gene. And you'll find that there's different ways to display these. Pack, squish, full. You can see what happens. Usually when you do full, I'm seeing the label here on the rhodopsin. This NCBI, this is a combination of ref sequence genes from basically the American database NCBI and ensemble built transcripts that are at ensemble in the EU. These are good. I actually like using the main now more than just the ref sequence curated. So you might want to just go with the main. Boom. And that's how you can get a picture and find your gene.